And with that, I'd like to introduce our final speaker, last but certainly not least, Dr. Mao, who is from the University of Vermont, and will be talking to us today about the little brain in the gut, although, correct me if I'm wrong, I do believe the big brain also will make a guest appearance at some point. So with that, Dr. Mao, thank you very much.
Thank you. I'm pleased to be here to preach the gospel of gut neurobiology this morning. Some of the things that I'll try to cover today include just a word or two about the organization of the autonomic nervous system and how I try to introduce it to medical and physical therapy students when I'm covering these topics. And then we'll talk about the organization of parasympathetic and sympathetic innervation of the gut and how long reflexes regulate upper GI function, but shorter intrinsic reflexes are largely responsible for controlling activity in the intestines. Then I'll show you an example of an experiment demonstrating very clearly how the central nervous system can override intestinal function, even though that intestinal function is controlled locally. And then finally, we'll talk about a couple of hot topics in enteric neurobiology. So in terms of organization of this system, as you know, the peripheral nervous system is divided into somatic and autonomic, and typically we see the autonomic nervous system being divided into parasympathetic and sympathetic divisions. Hopefully, by the time I'm done today, you'll be convinced that there should be a third box there labeled enteric division. So the autonomic nervous system differs from that somatic division in many ways, but one of the things that I try to emphasize when I'm teaching these systems to, let me see if there's another one here, nope, to the medical and PT students is that striated muscle is relaxed in the absence of neural input, and it contracts when it's exposed to acetylcholine. The target tissues that are innervated by autonomic neurons, including cardiac muscle, smooth muscle, and gland cells, have constitutive activity. So if you take a cardiac muscle cell out and put it and grow it in culture, you can see it contracting rhythmically. Smooth muscle cells generate tone on their own, and gland cells can secrete on their own. So rather than to cause or generate activity in these cells, the role of the autonomic nervous system is to regulate the activity in these cells, up and down regulate it. And that's why in most cases, with the exception of vascular smooth muscle, we often have dual innervation of a given tissue. And this is the heart as the example, where you have inhibitory input from the parasympathetic division and excitatory input from the sympathetic division, and the response in a given tissue depends on the neurotransmitter, the receptor, and the signaling pathways that are involved and what those signaling pathways are doing to ion channels. So if we take this pattern of innervation and overlay it on the gastrointestinal tract, this is the way Netter drew it, and it's, I think, pretty accurate depiction in some ways. The vagal preganglionic projections are shown here. As you know, the word vagus is from the Latin term vagabond, and this vagus nerve wanders down through the thorax and into the abdomen, and it's innervating much of the GI tract, about as far as the splenic flexure. From there, we have the sacral parasympathetics coming from the S2, 3, and 4 in human, coming out into the distal colon and sigmoid colon. With regard to sympathetic innervation, it's coming from thoracolumbar via pre-vertebral ganglia, the celiac ganglion, superior mesenteric and inferior mesenteric ganglion, and their projections follow the blood vessels. So here's the pattern for vagal innervation of the GI tract. It seems to go further than that splenic flexure, but this is the innervation, and the sacral preganglionics cover beyond that. But let's take a more careful look at the vagal input to the gut. So this is from work by Hans Rudolf Berthoud and Terry Powley. And what they were doing was they were labeling vagal efferent fibers and taking a careful look in the wall of the gut at where those efferent fibers were going. And what percent, this graph is showing the percentage of ganglia that receive vagal efferent input. And I should say this is my enteric ganglia. So just to show you what's going on here, up in the stomach, essentially 100% of the ganglia receive vagal efferent input. And if you look in these ganglia and you look at the labeled fibers in the neurons, it looks like essentially all of the gastric neurons have vagal efferent fibers associated with them. Gallbladder ganglia are likely to be the same. This wasn't looked at anatomically, but I looked at it electrophysiologically in my lab, and all gallbladder neurons receive vagal efferent input. The same is true of pancreatic ganglia. But as you can see here, as we work our way along the intestines, there's a rapid drop-off in the duodenum of the percentage of myenteric ganglia that are receiving vagal efferent input. And if you look at micrographs of these ganglia, you find that it's only a small subpopulation of the neurons that are receiving that input. So half or less than half of the ganglia receive vagal input, and in those ganglia that do receive it, half or less than half of the neurons are receiving that type of input. So these, from the terminology, are postganglionic neurons, but in the intestine, most of them do not receive preganglionic input. So this system is different. And if we talk about the submucosal ganglia, they don't receive vagal efferent input. They're on their own. So if we look at blood supply of the gut, this is the pattern of vascular perfusion from the celiac trunk, superior mesenteric and inferior mesenteric, and the pattern of innervation from those associated ganglia is the same. But let's look at the pattern of innervation of the GI tract from those ganglia. And this is a schematic from a review by John Furness, and it shows in the upper left-hand corner, this is a paravertebral ganglion, and then this represents one of these prevertebral ganglia. One type of innervation we have is vasoconstrictor innervation. So there are projections from prevertebral ganglia and paravertebral ganglia to blood vessels in the gut that cause vasoconstriction. We also have the ability to decrease motility, and the neurons that are doing that are not projecting to the smooth muscle, they're projecting to myenteric ganglia, where they largely have presynaptic inhibitory effect on synaptic communication within these myenteric ganglia using alpha-2 adrenal receptors. The way the sympathetic nervous system downregulates secretion in the gut is once again projecting to submucosal ganglia, not to the epithelium. And here we have inhibitory postsynaptic potentials and presynaptic inhibition in the submucosal ganglia. We also have innervation of immune tissues from these sympathetic neurons. But in terms of what we would have thought of as the classic example of sympathetic innervation of the gut, projections to smooth muscle and to epithelial cells, those projection patterns are very sparse. So this pattern of control is a little bit different. We also have neurons shown here in the gut that are projecting out to the sympathetic prevertebral ganglia. And they represent one of the major synaptic inputs or regulatory inputs to the prevertebral ganglia. It's a local loop between the enteric nervous system and the sympathetic ganglia. So if we look at the pattern of innervation, or a schematic, as I drew you for the heart, most of the neurons in the intestines don't receive parasympathetic preganglionic input. The target of the sympathetic division isn't the effector tissues, but it's rather the enteric nervous system. And the enteric nervous system actually sends projections to the sympathetic division to help it know when it should be downregulating gut activity. So this is a different system. And the idea that the innervation of the gut is different than other parts of our body is not necessarily new. Bayliss and Starling, in a Journal of Physiology paper that they published in 1899, proposed the law of the intestine, which states that local stimulation of the gut produces excitation above and inhibition below the excited spot, and these effects are dependent on activity of the local nervous mechanism. So what they appreciated at that time, 116 years ago, was that there's complex neural reflex circuitry that is housed entirely within the wall of the gut. That if you stimulate at one spot, you can get excitation or contraction above and relaxation below, and this is the underpinnings of peristalsis, and it's all within the gut. So this means you have to have some way to sense that stimulation. You have to be able to send that message upstream in the gut and cause contraction, excitatory motor neuron activity above. You have to send the message downstream, and you have to have inhibitory neurotransmission down below, all going on within the wall of the gut. And this was appreciated by John Langley when he wrote his big book, The Autonomic Nervous System, and he really laid out the organization of the autonomic nervous system as we know it, or maybe as we should know it. And as you can see here, he divided the autonomic nervous system into three divisions: the sympathetic, the parasympathetic, and the enteric division. And I think that's how it should be taught. So let's talk about the innervation of the gut. And as I mentioned early on, I usually talk about it as upper GI reflexes and then intestinal reflexes. And so in the case of the upper GI reflexes, we've got innervation, we've got sensory innervation or information coming from the vagus nerve or being generated in our cortex. And that information is passed to the vagal motor nucleus, and then we have output to the intestines, or to the gastrointestinal tract, that cause different things to occur. But let's talk for a moment about the afferent end of this reflex. We've got a lot of cells in the lining of our stomach and our intestines that are essentially acting as taste cells. These cells have microvilli with nutrient sensing receptors, acid sensor receptors. These can be activated, so they're actually like taste bud cells in the gut. As you know, their secretory vesicles are located on their basal surface, and when they secrete, their secretory compounds can enter the bloodstream, and they can activate receptors on nearby nerve fibers. And we have a lot of different types of cells that fit within this enteroendocrine cell category, and I'm not going to list them here, but we're learning to appreciate them more and more every year, and this is a really exciting field, I think, the field of taste in the gut. One of the recent findings along these lines, Roger Little's lab has shown that CCK-producing cells, this has been shown for somatostatin cells in the stomach, but Roger Little has shown recently that in the intestine, the CCK cells have these projections that he's referring to as neuropods that seem to be interfaces for neural input and for output from these cells to afferent nerve fibers. So this is like a kind of a synaptic type communication that these cells are capable of. So let's talk about types of signals that are being delivered by these cells to stimulate vagal afferent fibers. From the stomach, we have ghrelin and we have leptin, and also stretch receptors on the vagal afferents can activate vagal afferent input to the central nervous system. In the upper small intestine, three of the important signaling molecules include cholecystokinin, serotonin, and glucose-dependent insulinotropic peptide. I should mention the 5-HT3 receptor is listed there for serotonin. As you may or may not know, when individuals are suffering from nausea and emesis associated with chemotherapy, it's due to an overstimulation of the enterochromaffin cells and release of serotonin activating vagal 5-HT3 receptors. And this is why compounds like ondansetron, 5-HT3 receptor blockers, are now being used as such effective anti-nausea medications for people undergoing chemotherapy. Then as we get further along in the GI tract, a couple of the important signaling molecules to consider are GLP-1 and PYY. And there's been a vast increase in our understanding of these signaling molecules further along in the GI tract recently, in part because of bariatric surgery. So initially, bariatric surgery, the idea of it was that you would take away the accommodating ability of the stomach and send these calories through the intestines at a rate faster than we're able to digest and absorb them. And that probably is true to some extent. But what we've been learning is also these calories, now that these nutrients are getting to the distal ileum, they're activating increased release of GLP-1 and PYY. And this is doing a couple of things. The GLP-1 is acting hormonally to change our entire glucose handling ability. And so this is why people who undergo bariatric surgery are miraculously cured from their type 2 diabetes within a matter of days, and they have normalized glucose levels. Also, these compounds are acting on vagus nerves and changing satiety signaling in these individuals. And they even change their food preferences. So you have people going through these surgeries who are meat and potato people. All of a sudden, they're wanting to eat a salad, which is something that they never wanted to do before in their life. So this is, I just wanted to point out that this is a pretty exciting field. So some examples of some of these long reflexes that I'm talking about. One example is transient lower esophageal sphincter relaxations. I don't know if you've heard of these before, but this is kind of a form of burping. So stretching in the fundus, air in the fundus, leads to a transient relaxation of the lower esophageal sphincter, and that causes venting of air from the fundus. And this is an important reflex from the perspective of gastroesophageal reflux disease, the most common GI disorder. It seems to be an abnormal signaling of this TLESR reflex that is responsible for that, and these people have too many of these relaxations going on that is allowing refluxes to occur. And the pharma industry is interested in trying to get these under control and have been trying to aim at this with some of their meds. Another long reflex that occurs in the cephalic, oral, and gastric phases of digestion includes gastric accommodation, in which we have vagal projections to nitrogenous neurons in the stomach that cause relaxation of the proximal stomach. And then another type of reflex is the increase in antral pump activity that you see during the gastric phase of digestion. So I'm going to take a moment here to show you an example of that. And unfortunately, one of these Mac PC things, I've got to go to my movie thing here. Whoa. Let's see here. Okay, so this is, I'm looking for this sound. We'll do it without sound. But I wanted to show this to you as an example. These videos are available online. They were produced by a Professor Ehrlein at University of Hohenheim in Germany, along with Michael Schiemann, who is an enteric neurobiologist who worked in his lab. These are available online. There are a lot of gastric physiology videos and a lot of intestinal motility videos. There's a real nice video showing emesis occurring, a dog vomiting after it's been given apomorphine. But as you'll see here,
into the gastric body and the antrum. The location of the pylorus and the duodenum are also indicated.
I think they're very effective for showing students how these things are working.
Represent the gastric pump.
So here you can see the proximal stomach is just sitting there, not doing anything.
The peristaltic waves originate at the gastric body and propagate towards the pylorus. When the peristaltic wave reaches the antrum, the constriction of the wave becomes deeper. At the antrum, 3 parts can be differentiated.
Okay, we won't go into that, but neurons in this area of the stomach, kind of the middle corpus or body of the stomach, are activating the pacemaker activity in interstitial cells of Cajal, which in turn are involved in starting this antral pump or this antral pump ring of constriction that rolls its way towards the pylorus. And so we can increase the rate and the strength of these events by increasing vagal input to gastric ganglia and in turn neuromuscular or muscular activity. Okay, another example of a long reflex is acid secretion. The major driving force for acid secretion is vagal and local gastric neural input to the stomach. So it's working in three different ways. One way is directly on this is a gastric neuron now receiving vagal input. It releases acetylcholine onto muscarinic receptors on the parietal cells. It causes histamine release from enterochromaffin-like cells in the region, and it also can promote gastrin secretion from G cells, another of these types of enteroendocrine cells. The gastrin works through a hormonal mechanism to increase acid secretion and also to activate ECL activity. Before we had proton pump inhibitors and histamine blockers, if somebody had really bad gastric ulcers, one of the treatments for this was a bilateral vagotomy. And this is also obviously before we knew about Helicobacter pylori. So parietal cell secretion is a neural event. Also, chief cell secretion involves stimulus being acid in the lumen, activating local enteric reflexes and vagal reflexes that cause muscarinic receptor activation on the chief cells, and they're releasing lipase and pepsinogen, and the acid and the pepsin converts pepsinogen to pepsin from there. So we also have coordination of upper GI motor activity once the gastric contents are entering the small intestine, and they're activating eye cells that release cholecystokinin in response to proteins and fats. And this CCK works through several different mechanisms to regulate upper GI motility. One thing it does is it acts hormonally to increase gallbladder contraction. So CCK, I believe, was the second hormone discovered. It was an American Journal of Physiology paper by Ivy and Goldberg in 1928, and they hooked up the vascular system of two dogs and showed that when they fed one of the dogs, the other dog's gallbladder would contract, or if they infused CCK into the one dog, the other dog's gallbladder would contract. We also have local enteric reflexes that can lead to sphincter of Oddi relaxation, and then we have vagal reflexes that are slowing down the stomach activity. So we're further relaxing the fundus, we're slowing down the antral pump activity. So what we're saying, what the duodenum is saying to the stomach is, okay, we've got a lot of nutrients we've got to work on. We need to slow down gastric emptying so we can digest and absorb these calories. That reflex also contributes to gallbladder contraction and pancreatic secretion. And the net result is decreased gastric emptying and increased bile flow. Within the pancreas, we have long neural reflexes that are associated with the acinar cell secretion. Once again, this is CCK acting on vagal afferents. CCK itself, but probably mainly acetylcholine coming from pancreatic ganglion cells, activates muscarinic receptors on the acinar cells to increase acinar secretion. And then we have secretin, the first hormone discovered, entering the circulation and acting entirely through a hormonal mechanism to increase duct cell secretion, and that duct cell secretion flushes the pancreatic enzymes into the small intestine. So those are examples of long reflexes. So these are all upper GI tract, and they're involved in the initial phases of the digestive process. From here we get down into the small intestines, and this is where everything is regulated a bit more locally. So this is a schematic diagram by Furness and Costa of the enteric nervous system. You can see layers of the gut peeled away here. One thing that this shows, this diagram shows both small intestine on the left, and then on the right side you can see colon because we don't have any villi here. In the submucosal layer, we've got a ganglionated plexus that's largely involved in regulating secretion in the gut and also regulating blood flow, as I'll tell you about in a minute. And then between the circular and longitudinal muscle layers, we have the myenteric plexus. So if you look at this in histology class, it's not very impressive because you're looking at the enteric nervous system on edge. On the other hand, if you have an opportunity to look at it en face, you begin to see that there's this vast sea of ganglia that stretches from the distal esophagus all the way to the anal canal. And there are a lot of neurons in this system. Many of you have probably heard this fun fact that there are more neurons in the GI tract than there are in the entire spinal cord, over 100 million. So this shows the myenteric plexus in the guinea pig. So let's focus in on a little section of one of these ganglia and look at it at the electron microscopic level. And you'll see another interesting feature of the enteric nervous system. So this is the area of this micrograph that shows the myenteric ganglion. To the left, there's like a basement membrane, and then you see smooth muscle there. But if we look at the ultrastructural features of the ganglion, and you focus on this box, you see something that's very peculiar with knowing that this is peripheral nervous system. These are unmyelinated axons that are running right next to one another. Normally, you only see that in the central nervous system. In the peripheral nervous system, individual unmyelinated axons are typically wrapped by Schwann cell sheaths. There are a lot of, so the ultrastructurally, the enteric nervous system looks more like the CNS than the PNS. Also, there's quite a bit of neuronal diversity in this system. There's diversity of morphological characteristics of these neurons. There's diversity of electrical and synaptic activity in these neurons. Chemical coding patterns. This micrograph shows a lot of different types of neurons. I won't go into, what we're showing there. But also, different subpopulations of these neurons have different projection patterns. So it took basically from 1899, when Bayliss and Starling were doing their work, all the way to the late 80s and through the 90s for we as enteric neurobiologists to figure out which neurons were doing what. We knew that circuitry existed for peristalsis, but we were looking at this sea of ganglia and this sea of neurons and not knowing what was what. But by combining morphological techniques, intracellular injection with dyes, with knowledge of the electrical properties of those neurons, and then seeing where their axons project, and then doing immunostaining, we slowly were able to put together a picture of the different types of neurons that are found within the intestines. And with this combined knowledge of these neurons, we could start assigning functions to the different types of neurons. So we have intrinsic sensory neurons, these neurons that are purple and red. Intrinsic sensory neurons, 100% of them project to the mucosa, where they're able to receive signals from those endocrine cells. We have a lot of interneurons. Some are projecting upstream in the gut, some of them are projecting downstream in the gut. And then we have motor neurons to the muscle, motor neurons to the epithelial cells, and also motor neurons to blood vessels. There are a lot of different neurotransmitters in this enteric nervous system. Every class of neurotransmitter can be found here, and there's quite an array and a lot of co-expression of these neurotransmitters. Some of those that we have assigned pretty important functions to are shown in green or red, green for excitatory, red for inhibitory. Apologies to those of you who are red-green colorblind out there. And ATP, it can be both excitatory and inhibitory in this system. Along with all of this vast array of neurotransmitters, the enteric nervous system is a pharmacologist's playground with regard to the number of receptors that are here. This is a map from a schematic diagram from a review article that we published recently in Nature Reviews GI, showing the distribution of serotonin receptors along the reflex circuitry of the gut. So this is different types of reflexes here. You can see serotonin receptors on the afferent terminals, on cell bodies, and on presynaptically on nerve terminals. So this is a rich array of different types of receptors along this system. We can't talk about the enteric nervous system in 2015 without acknowledging enteric glia. There are a lot of presentations at this meeting by Brian Gulbranson's lab, about enteric glia, and he's giving a talk tomorrow afternoon, I believe. This is staining of a guinea pig myenteric ganglion stained for S100, showing the glial cells here. These cells in the gut are positive for glial fibrillary acidic protein, which is known to be synthesized by astrocytes in the central nervous system. The first efforts to see what GFAP does in astrocytes was a knockout mouse. They looked in the brain, didn't see anything going on. These animals were dying young. They necropsied them, and they found that all of these animals had widespread enteritis when they knocked out GFAP. This is the same micrograph showing the glia wrapping around the neurons. But now we know that there are enteric glia out in the epithelial layer of the gut, or in the mucosal layer of the gut, just underneath the epithelial cells. And they seem to be integral for maintaining epithelial barrier function. We're also learning a lot of other functions of these enteric glia in terms of neuronal growth and survival, and also neuronal communication. So this is really an emerging field. So the most unique feature of the enteric nervous system is the presence of intrinsic reflex circuitry. This is the only part of our body where we have sensory neurons that are located outside of the dorsal root ganglia and cranial nerve ganglia. And this is an example of one of these reflexes. So this is layers of the gut, orals to the left, aborals to the right. Stimulation, and this can be in the form of chemical stimulation and or stretch, leads to direct stretch activation of neurons and also release of compounds like serotonin from enteroendocrine cells that activate the intrinsic sensory neurons, once again, all of which project to the mucosa. These intrinsic sensory neurons activate ascending interneurons that selectively synapse on excitatory motor neurons and cause a contraction, primarily cholinergic contraction above the level of the stimulation. Those intrinsic sensory neurons also activate a descending limb of this pathway that involves inhibitory motor neurons that release purines such as ATP and beta-NAD, and also nitric oxide to cause a relaxation downstream. Contraction above, relaxation below generates a pressure gradient. The luminal contents move along the gut, and then it repeats itself because this circuitry is omnipresent along the intestines, along the small intestine and the colon. And so I brought some movies along here to show you the enteric nervous system in action. This is a segment of guinea pig distal colon, and what we've done is we've taken guinea pig fecal pellets and coated them with nail polish. This is the first job for new undergrads in my laboratory to really test how interested they are in science. But you can see that this pellet is moving in a pretty linear fashion down along that segment of guinea pig distal colon, and it involves that reflex that I just showed you. Contraction above, relaxation below. And we know that this, like Bayliss and Starling were describing, involves the activity of the local neural mechanism, because if we block action potentials, if we block voltage-activated sodium channels and put the pellet in the same segment, you can see some myogenic contractions, but we're no longer able to generate that pressure gradient, and so the pellet just sits in that same spot. I'll also mention, I'm not going to get into the data here, but we've shown that if we activate all of the neurons in the enteric nervous system, the pellet doesn't move. We wondered if it would shoot across the room, and we call this because we do a lot of inflammation-induced neuroplasticity work in my lab, where motility is disrupted even though neuronal excitability is increased. And we think of this as attention deficit disorder in the enteric nervous system. So let's see here. Okay. So I just want to take a moment to talk just in passing about how these neuromuscular signals are processed. So there is like a neuromuscular motor unit in this system, and it involves interstitial cells. So the cells in red here are stained for KIT protein, and these are interstitial cells of Cajal. This is another one of these things that we're spending a lot of time studying these days, that Cajal described 110 or so years ago. But these are interstitial cells of Cajal. They form a network in this system. So those are the pacemaker interstitial cells. There are also cells shown in red and green here that are interstitial cells that seem to mediate neuromuscular transmission in this system. So the red cell represents an interstitial cell of Cajal, and it's mediating cholinergic neuromuscular transmission and also nitroergic inhibitory neuromuscular transmission. And it's just in the last 5 years or so that it has been discovered that another type of interstitial cell that had been invisible to us and there all along, this PDGRF alpha cell, mediates purinergic inhibitory neuromuscular transmission. These cells are full of purinergic P2Y1 receptors, and they're full of SK3 potassium channels, voltage-activated potassium channels. So when the purinergic receptors are activated in these cells, they dive towards EK, and that hyperpolarization spreads through the smooth muscle syncytium. So that's how neuromuscular transmission seems to occur in this system. There are other intrinsic reflexes in the gut. I mentioned to you early on that submucosal ganglia do not receive vagal efferent input. These ganglia are on their own. So there we have basically sensory neurons and motor neurons in these ganglia. The intrinsic sensory neuron is responding once again to release of serotonin motor neurons that release compounds like acetylcholine onto muscarinic receptors, increasing the open state probability of the chloride channels in those epithelial cells. It's probably the same motor neurons that are causing vasodilation in this system. So submucosal neurons can cause vasodilation. So this is local vasodilation. When we're processing a meal, we want more fluid coming to the gut so we can secrete water, and we also want that vascular dilation because we're absorbing a lot of nutrients into the vessels in this area. So we are increasing the blood flow in the area where digestion is occurring through these local reflex mechanisms. There are also, up in the area of the stomach and duodenum, there are also these local reflexes that appear to exist that we don't know a whole lot about, but they're kind of interesting. And we've studied a couple of these in my lab. One of these is a local neurons in the duodenum project to the sphincter, to the pyloric sphincter. So this could be a way of regulating gastric emptying from the duodenum in addition to those circuits that I was talking about earlier. There are also neurons in the duodenum that project to the gallbladder. This is something that I discovered when I was a postdoc in Mike Gershon's lab. There are also neurons in the duodenum that project to pancreatic ganglia and can increase pancreatic secretion. And finally, there are neurons in the duodenum that project to the sphincter of Oddi. And this was a lot of this work was done in my lab. I had a grad student that showed that these neurons are intrinsic sensory neurons that are sensitive to CCK and project into the sphincter of Oddi. So there's kind of a lot of local control going on here. So these in the intestines, activity, these functions, fundamental functions, motility, secretion, and vasodilation are controlled by local neural mechanisms. But it's important to note that the big brain can override the little brain in the gut. This is a slide that's depicting an experiment that was done by Professor Tom Almy, a gastroenterologist at Dartmouth College. This, as you can see in the lower right, this was published in 1947. And Almy was interested in the relationship between stress and GI function. And he invented this contraption that he has on his head there, which is this metal crown with these screws that can be tightened. And so what he would do, he would recruit one of the better experimental models at the time, medical students, into his lab, and he would put this thing on them, and then he would take a rigid sigmoidoscope and put it up into their rectum. Now, they didn't have these really nice, small, flexible fiber optic systems at the time. This was kind of like a small telephone pole. So Almy would put this up into their rectum, and he would look at the amount of motility going on as reflection of motor activity, and he would look at how red the mucosa was as a reflection of blood flow to the mucosa. And as I mentioned to you, both of these are regulated by local neural mechanisms. He had these people put their hand in ice water and showed that they reported pain, but they weren't seeing any changes in their engorgement of the bowel or contractile state of the bowel. So then he would say, okay, I would like for you to tighten those screws on your head as tight as possible. And so they would, crank on these things, do it as well as, and Almi would say, so that's all you can do there? And he'd say, yeah, doctor, that's as tight as I can get them. And so this guy's name, let's say his name was Paul Smith. So he'd say, Paul, you've got a classmate named Mark O'Reilly, right? Yeah. And Tom Almi says, and you and Mark are applying for that same residency at Beth Israel, right? Mark also asked me to write a letter of recommendation for him, and he was in here the other day, and I have to say he tightened that crown a lot more tightly than you seem to be tightening it here. And at that point, with this threat of not getting the good letter for Beth Israel, all of a sudden motor activity increased, and also there was vasodilation. So he published this in 1947. And there was a lot, it caused a bit of a stir, but there were still people saying there's a pain component to this. This is not just stress, this is pain. So Almi came up with another brilliant idea. He had to wait a couple years, I think, for the med students to graduate and, word of mouth to fall away. And so what he did, got that sigmoidoscope out again, put it up in the rectum, and did kind of like just baseline activity measurements. And then Dr. Almi said to his nurse, I don't think I've ever seen anything like this. I'm going to have to take some samples. And could you call surgery and call pathology? Let's have them do frozen sections right away. And so he even had a little container, and he had like a little hunk of potato that just in the visual range of this medical student, he would drop this piece of potato into a sample vial and have the nurse run off with it. And so when this was going on, this discovery of a rectal carcinoma, you can see that this is a stress response here. And it fell away. Now, this was before IRB approval, right? But it still, I think, serves as a good example of what's going on here. But another feature of this that I want to mention to you is that we really don't know what is going on here. The first person who I saw show this slide was Jack Wood, who many of you might know, very famous enteric neurobiologist now at Ohio State University. When I was putting this talk together, I've been using this, but I called him and I said, Jack, let's talk about that experiment. Neural pathways don't completely explain this, and stress hormones acting on the enteric nervous system don't completely explain it. And what Jack thinks is going on is that it might be stress hormones possibly released, locally and or through the HPA axis acting on mast cells in the region. And we know that mast cells, back to neuroimmune interactions, mast cell degranulation is really effective at flushing our GI tract of toxins and infectious agents if we need to do that, because it causes a really big simultaneous secretory event and motor activity, and it just pushes everything along, and it also goes along with vasodilation. So that there might be a mast cell component to this. So any self-respecting nervous system has disorders associated with it, and we now know that there are a lot of motility disorders that are associated with changes in enteric function. We also know that IBD leads to neuroplastic changes that are responsible for altered motility, secretion, and sensitivity in the gut. And also, I have a question mark with IBS, but it's becoming clear that there are changes in the wall of the gut in irritable bowel syndrome. In my lab, we've shown that there's altered serotonin signaling in the gut. Others have shown a slightly elevated immune state in the gut. Those are probably interrelated. This is likely leading to visceral hypersensitivity and altered motor activity in the system. Before I leave this, I want to just show you how these sensory signals are processed. We talked about the intrinsic afferents. The vagal afferents seem to be involved primarily in homeostatic and digestive reflexes, a lot of satiety signaling here. But when we get to pain and discomfort, these signals are being processed by way of the spinal afferents. So if you're trying to affect visceral hypersensitivity, it's the spinal afferents and their communication that you want to work on. 5-HT3 antagonists seem to inhibit these signals, and somehow, and we don't understand it, but 5-HT4 agonists also seem to dampen visceral sensitivity through some sort of peripheral mechanism. So in closing, I just want to talk to you about a couple of things that are going on in this system that are exciting. Not work that I'm doing in my lab, or we're doing in my lab, but some things of interest. And one was really nicely described by Dr. Aboud in the second talk in this session, and that has to do with vagal stimulation. And so this is a list of things that vagal stimulation has been shown to do. It's pretty much everything but peace in the Middle East, I think. So rheumatoid arthritis, inflammatory bowel disease, asthma, diabetes, obesity, migraine, seizures, and depression. So these are some things that we don't completely understand how this is going on, but there might be some cues in the next little section that I'll give of the talk. But the pathways involved in this vagal anti-inflammatory pathway to the gut are being worked out. So with regard to rheumatoid arthritis, it's that stuff that the pathway that Dr. Aboud mentioned that Kevin Tracy has been working on and Robin McCallum. The details of this circuitry I don't think have been clearly worked out. This shows a vagal input to the celiac ganglion. I don't know if that really happens, and then celiac neurons projecting to the spleen. And here's where we have those alpha-7 adrenoreceptors, or I'm sorry, nicotinic receptors on the macrophages. And we have T cells in this model in the spleen releasing the acetylcholine. Work by Guy Buxtein and his colleagues at University of Leuven in Belgium have been studying the GI component of this reflex, and they have found that vagal efferent fibers that are going down to the intestine, which I mentioned to you are on the sparse side, seem to cause release of acetylcholine from myenteric neurons, and it's this local myenteric neuron input to the macrophages and their alpha-7 nicotinic receptors that seems to be involved in this intestinal vagal anti-inflammatory pathway. But this is kind of a pretty cool story, and I think if we can take advantage of this, this might represent a novel approach to treating IBD and other inflammatory disorders of the GI tract. You can't give a talk in 2015 without the microbiome coming up, especially if it's a gut talk. But one of the most interesting aspects of the huge deluge of information that we have about the microbiome has to do with the microbiome and the gut-brain axis. There are a couple of labs. One at John Cryan's group at Cork University Medical Center in Cork, Ireland, and also the group at McMaster headed by Steve Collins, Elena Verdu, and Pramuk Burkic, who's shown in this picture here, have done some really elegant work showing that if you change the microbiome, you can change animal behavior and brain neurochemistry. And one thing they've shown is that exploratory behavior is increased in germ-free mice. So you take the biome away from a bulb C mice, which are pretty timid animals, and now they become more exploratory. This is a step-down paradigm and also time in light. This is, I think, a plus maze. But even more interesting than that, in this paper by Premack-Burkick, what they showed was if they took two different strains of mice that have different behaviors. NIH Swiss mice are pretty exploratory and assertive. Bulb C mice, like I said, are pretty timid. But if you take the microbiome from NIH Swiss mice and you put it in germ-free bulb C mice, they become more exploratory. And if you take bugs from bulb C mice and put it into NIH Swiss germ-free mice, they become less exploratory. So there's something going on here. And not only that, but it seems to involve a critical period. So there's this period during development, so it's sometime before 10 weeks of age that you seem to have to make these changes in the microbiome to see these changes in behavior. So this is kind of an interesting part of this. How is this information communicated? Some of these changes are blocked by vagotomy, others aren't. With regard to those that are mediated by the vagus nerve, how does the information get from the bugs to the vagus nerve? Work by Wolfgang Kunze, also at McMaster University, has shown that there seems to be a nicotinic synapse from myenteric neurons onto vagal afferents that is critical for this vagal signaling from the gut in response to bugs in the lumen. So we're kind of redrawing our autonomic circuit diagrams here, both with regard to how the autonomic nervous system contributes to organ function and in inflammation, and also how signals from the lumen of the gut, from the bacteria, are getting to the central nervous system. So in summary, one thing I want to try to drive home here is that the enteric nervous system is a distinct division of the autonomic nervous system that exhibits unique neuronal circuitry. Long reflexes regulate upper GI tract functions, while local reflexes regulate intestinal functions. But we know from Almi's work and many others that the big brain can override the little brain in the gut. And finally, advances in our understanding of the enteric nervous system could improve treatments for many GI disorders, including IBD and IBS. So thanks for your attention.